Welcome everyone, my name is George. I've been a PhD student at the Adaptive Genomics group led by Professor Michael Hofreiter at Potsdam University, and have been mainly working on ancient DNA using mammalian species such as beavers, brown bears, cave bears and mammals. Today I would like to give you a presentation which has a long title on the how and why of different DNA extractions and library building approaches. This is about extraction and library protocols we often use in ancient DNA research, the reasons why scientists had to develop new methods, and how these methods are implemented in the lab. So first let me give you an overview about the content of this presentation. I will begin by talking about the source of DNA, where do we actually find it, what makes DNA ancient, its properties, and what are the usual problems we encounter when working on ancient DNA samples. Then I will move on to the extraction, discussing some common issues one must take into consideration before the actual extraction step. I will continue with the extraction method design, and in the end of this section I will provide some empirical evidence on the method's performance. The next part will be about DNA library construction. Here I will address the details of the so-called single-strand library protocol, how is it designed. I will also discuss its advantages compared to other library building methods, and lastly present some results that compare yields between different library building protocols. The last part will be a summary of the main points of this talk, an overview of the achievements in the field of ancient DNA research, and some personal views about future perspectives. So today we use all kinds of sources for retrieving DNA. It all started in 1984 with this species. This is the quagga, where they use the dried muscle sample to sequence part of its mitochondrial genome, and since then probably the majority of studies have been using old bones and teeth for recovering ancient DNA. Of course there have been other substrates used, such as hair, skin, cells, or whole body parts in case of insects. Of course ancient DNA can be applied in plants. For example, we can extract ancient DNA from seeds and pollen. And there have been also studies using historical artifacts and to extract DNA, such as pottery or skin parchment. And there is also an increasing interest in retrieving ancient DNA from sediments. So we can find ancient DNA pretty much in every old, so to speak, sample. The number of sources has increased with time, and this also expanded the applications of ancient DNA in other fields such as archaeology, paleoecology, forensics, etc. But what does it really make ancient DNA? What features does ancient DNA has? So we recognize three main characteristics. The first one is that ancient DNA is short. So if we plot the DNA fragment length of an ancient sample against their frequency, we usually get this characteristic distribution. So here on the x-axis in the DNA is the DNA fragment length in terms of base pairs. And on the y-axis we get the number of fragments corresponding to its length. So most fragments are within the 20 to 50 base pair range, and there's a very steep decline if we move to longer fragments. This pattern is the result of fragmentation, DNA fragmentation, where basically a species dies, its DNA repair mechanism ceases and cell membranes break down, and DNA is exposed to all nuclease activity and other kinds of environmental factors. And with time it degrades into shorter and shorter pieces of DNA. So the mechanism for that is basically a two-step process. First we have hydrolytic effects on the sugar-base bond. The base is removed. So in this case we have the removal of guanine, a purine. So we have a depurination. And then through beta elimination, the phosphate group at the free end of the DNA molecule is removed, leading to DNA fragmentation. Basically the backbone of the DNA breaks, and this breakage has been shown to to occur more often at sites adjacent to purines, meaning A's and G's. There are now good evidence to support that the severity of this process depends largely on temperature, among other environmental factors. And based on that, in 2014 Hofreiter et al. And built a model for the expected survival of DNA, taking into account three factors: temperature, sampling site, and length of fragments. So on the maps here we show how much DNA in terms of percentage is expected to survive after 10,000 years of fragmentation process in different site types, whether these will be caves or deep burial sites named after deep on the map, and or open sites, surface, as they depicted on the map. So basically their prediction is quite close to what we find from empirical studies, and what we can infer from these maps is basically three things. First, the longer DNA fragments have a lesser chance of survival outside of the permafrost region, while shorter fragments are also expected to be found in separate environments. The maps also stress the importance about deep burial sites and caves that have the potential for greater survival rates for ancient DNA than compared to open sites. And lastly, the most obvious pattern, of course, is that the tropical and desert regions, in tropical and desert regions, the percentage of DNA surviving is very low for any DNA fragment length tested. So essentially, these maps give an impression of what kind of problems we're dealing with in ancient DNA research, and how difficult it is to find DNA outside of the optimal sites, so to speak, whether these are caves or permafrost. So we talk about fragmentation. A second ancient DNA characteristic is the so-called miscoding lesions. Again, through hydrolytic effects, DNA bases are deaminated. This typically happens in cytosines, where deamination will convert them into uracils. And this potentially creates problems. This damage pattern creates problems during elongation steps, because when polymerase reaches the modified cytosine site, instead of adding a G base, incorporates an A, and this can lead to erroneous codes when performing sequencing, and potentially some mapping problems, since it can add more mismatches to the ensign read compared to the reference genome. So deaminations can take place at any site along the ancient DNA molecule, but they occur with much higher frequency at the ends of the fragments, and this is evident from the increased frequency of thymine bases at the ends of the ancient DNA reads in sites where the reference carries a cytosine base. So we have these C2T substitution patterns due to miscoding lesions. This can optionally be eliminated by chemical treatment in ancient DNA extracts, so that removes the uracils and prevents this problem. But researchers often prefer to retain them, because they also act as an authenticating indicator for ancient DNA, for the presence of ancient DNA in their sample. So the third characteristic of ancient DNA has to do with the low percentage within the total DNA extracted from a given sample. So when we extract DNA from an old sample, that contains, of course, the DNA of the actual species, which means the endogenous DNA, and all kinds of other DNA fragments from microbes, humans, etc. So these pie charts taken from Newman et al. Indicate the usual case in terms of endogenous content. So we usually find between 1 and at best cases like 6%. Of course, there are exceptions to this, like some from the permafrost region can give much higher endogenous content values, but everything else is pretty much contaminants, from microbia to humans to who knows what. So this is basically the most usual case in the samples that we deal with. And if we were planning, for example, to sequence a sample looking like that, to do what we call shotgun sequencing, to sequence the whole genome, that would, of course, cost us a lot of money, because that would mean that we would also sequence a lot of fragments that would be like of exogenous origin. So to sum this up, we have limitations for research across space and time due to fragmentation, meaning that we can usually hope to find fairly well-preserved samples from optimal regions, and the older the sample, the harder it is to find DNA in it. So fragmentation also causes problems with our extraction library building methods, which we will discuss later. We have potential problems when sequencing for erroneous base calls due to miscoding lesions, and we have also the problem of increasing costs from ancient DNA sequencing, for ancient DNA sequencing, due to the very low endogenous content that we usually find in ancient samples. So how do we overcome these problems? First, let's consider the low endogenous content problem. So samples are usually taken by museum collections, or ideally the case would be to take them straight from the excavation site and bring them in the lab. So you can imagine that a sample might have been buried for thousands of years and being handled by a number of people before ending up in a collection probably, and then we have it in our hands and we try to sample it and get DNA out of it. So the least we can expect is that this has been heavily contaminated, and the last thing we want is to contaminate it further. So what we normally do is take precautions, use protective gear, so we'll take in the lab and sample it there under stellar conditions optimally. Sometimes this is not possible, and of course we have to go on site at a museum or an office to do the sampling. But for a long time, scientists probably worked with what was available. So I guess rare samples or valuable samples would not be offered so easily, because of course there's a destructive process in the sampling where we use drills usually and destroy many of the characters of the original sample. So if I was, for example, to choose between two samples, I would probably pick up the one that looks less degraded, but this is not like a good criterion. A safe bet for preservation is comes from a study which suggests that the petrous bone in humans and likely other mammals contains very high endogenous content samples. So this is the petrous is the densest bone, in the human body. That's its name. And from the following graph that I will present you, you can see the amount of endogenous DNA in different samples. So we have humans and different species of bear, brown bears and cave bears. So these are all petrous samples. On the x-axis you have the different individuals, and on the y-axis you have the percentage of endogenous DNA. So by the look of it, you can see that most samples have above 10%, and they reach up to 75%, which is quite impressive actually. And just by this graph, it can tell you that how crucial it is to get the right sample for the job. So just by selecting the right sample, you almost secure that you will have really high endogenous content without doing anything else. Just to make things more specific, a study from Pinasi et al. Addressed the importance of selecting the right part from the petrous bone. So they divided it in 3 different regions marked as A, B, and C on the picture shown here. And they basically found that the piece that had the highest content was the C, the center one, and actually had like 65 times more DNA than the B, and 177 times more than the A part. And as one would expect, the C part was basically the densest part of the petrous bone. But of course, one cannot really access only petruses, and probably the next good alternative would be teeth, from what we know from the literature. But since the density of the bone has been addressed as an important factor, in our group there has been a study by Alberti et al. And they use CT scanning. This can recognize denser parts of the bone and less dense parts. So, and they use both petrous bones and long bones. So this in this study, on the right-hand side graph, we can see a clear pattern with the blue bars for each of the samples. These are the hard parts, and on the y-axis we have the endogenous content expressed as percentage of ridge mapping to the reference. So we can see a clear pattern that all samples that have been taken from the hard part of the bone end up having higher content in endogenous DNA, which is essentially very good to know, a very good thing to consider as a method. I mean, if you have access to a CD scanner, of course. So if none of the above applies, so if you don't have like a CD scanner where you can check the samples, or you don't have access to good samples such as petrous bones or teeth, one can consider doing some pre-extraction steps. So basically treating the samples with some sort of chemical. Usually people use bleach, or perform a pre-digestion step with a lysis buffer, and where after performing a first step where the first extracted elements which are expected to contain the most contaminants are removed, and then the rest is digested further. So these methods, of course, can be combined and can potentially have like a positive effect in increasing the endogenous content, but the results are quite variable among samples and sites. In a study we performed in our lab in Potsdam, we tested different concentrations against the different concentrations of bleach solution and against the pre-digestion method, and we had also a control. So we do find that we have up to ninefold increase in endogenous content, but for the treated samples, but at the same time we have a massive loss in relative DNA quantity. So we see the bars indicate the relative DNA quantity, while the horizontal marks indicate the endogenous content increase. So you have an increase in the endogenous content. The maximum was the 0.5% bleach treatment, but for all treatments against the untreated sample, we see a massive drop in DNA quantity. So there seems to be a trade-off there. So it is something to be applied with caution, I would say. So we talked about methods to basically ensure that you will have increased or with either pre-extraction method chemically increase the endogenous content of a sample. So we should move on to the extraction method. So basically, over the years there have been two major methods for extracting DNA. One has been described by Roland and Hofreiter, and another, which is basically a modification of that, was proposed by Dabney in 2013. Dabney et al. Sorry. The two methods are more or less similar with small differences. Basically, both include the digestion stage, where you have the sample and the lysis buffer, and then the digested sample is mixed with a binding buffer that's filtered through a column with silica filter. The DNA binds on the silica, washed to remove all excess of salts, and then eluted to get the final DNA extract. So the main difference between the two methods is that in the method proposed by Dabney et al., the binding buffer has a different consistency and a much larger volume. And for that reason, it uses a custom-made column for the DNA binding step, which I will show you on the next slide. This was essentially an optimization in an attempt to compensate for the potential loss of small fragments, which are expected, of course, to be found in degraded samples. So this is how the binding sort of device looks like. So in order to accommodate the large binding buffer volume, you use a reservoir where you load the binding buffer along with a digested sample, and you attach a silica filter on it, and this whole thing goes into a Falcon tube in order to be able to perform centrifugation. So after that, the DNA is bound. When you centrifuge it, the DNA is bound on the silica, and then you can elute it in the final solution after the washing steps. So how did this proposed method by Dabney perform compared to the other one? So in a initial test they did with artificial DNA fragments of variable size, they saw a clear pattern. With the second method, they were able to retrieve the one that is referred as optimized on the graph, you can see that you get a much higher rate of fraction of recovered short fragments for 35, 50, and also 75 base pairs, basically for all fragments. So this graph shows that this optimized protocol has the potential for recovering much shorter fragments compared to the previous method. And I would like here to show you another graph from a study done by my colleague Axel Barlow. So he has further investigated this by estimating the total amount of DNA in terms of nanograms from different extraction methods. So here they compared 3 instead of 2 methods. One is the original Roland and Hofreiter method, the other one is Damne et al., and then the third is some sort of hybrid between those 2 methods. The take home message from this is that in all samples tested, from shown on the x-axis, the Damne protocol, which is depicted with the black bars, yields much higher amounts of DNA from ancient samples of cave bears and brown bears that have been tested here. So except from one sample of the HV75 where it seems that it has equal yields with a combined method. So in general, Damne method seems to be more fitted for applications where shorter fragments are expected. So could say that it would be more suitable for specimen sample outside of the optimal preservation sites, where you would find where you would expect the degradation of DNA to be greater. And this is exactly why it was designed, actually. So I will show you more graphs about the effect on the fragment length distribution by this extraction method, and but we'll talk first a little bit about the library protocol. So we'll move on to the library section. So the DNA library has a basic structure. So you start with a DNA molecule, 2-stranded, double-stranded. You fragment it if it's modern. If it's ancient, you just leave it as this because it's already fragmented. You adapt, you ligate adapters, and then these adapters have priming sites for the sequencing primers and potentially barcodes to distinguish between samples when sequencing multiple samples at the same time. So under the same idea here, with the extraction methods, there have been like 2 widely used library construction protocols in ancient DNA, and one has been optimized specifically to deal with problems arising from ancient DNA properties. So the most commonly used library protocol is called double-strand library. Very briefly, its molecule it starts with double-strand molecule, and its molecule is treated so that it goes under a blunt end repair, so the overhangs are eliminated, and at the same time adapters are attached at both ends, which are filled in at the next step, and the next step is the indexing PCR, where you can use the library after that for the sequencing. So basically the conversion of DNA library is done on the 2-stranded molecule. In contrast to that, the single-strand library starts, of course, with a double-stranded molecule, and first you denaturate the library, and you get 2 single strands. So each single strand gets an adapter ligated at the free end, and the adapter at the edge has a biotin which can bind on magnetic beads, so it immobilizes the whole strand. And as it is immobilized, then you can start filling in the rest of the complementary sequence. And then from there, when the sequence is filled, you can, similarly to the double-stranded protocol, end ligate another adapter on the other side. And then lastly, you can do the indexing step. So at this point, I would like to mention that prior to the first step, optionally, you can use uracil sites, something I forgot. You can use treatment for the uracil sites, something I forgot to mention. So you can eliminate all the uracils that are usually found due to deamination in ancient DNA molecules. But as I said, most people just prefer to preserve them so that they can use that for authentication of ancient DNA molecules in their final data. So by looking at this and comparing to the double-stranded protocol, one could easily ask, like, why should I do this? Because that looks quite more complicated, and I can tell you for sure that it is actually. It's quite laborious, and compared to the double-stranded protocol, is much more expensive. So why should we bother doing this instead of the other? So the answer comes from the empirical evidence. So again, I'll show you some results that we had from internal work in our lab. So again, it's Axel Barlow study testing different extraction library protocols. So the graph shows here the conversion rate, basically how many molecules in the extract are converted into library. So the bars above value 1 on the y-axis indicate greater conversion rate for single-stranded molecules against double-stranded molecules, and the ones below that face below 1 indicate the opposite. So what we see here is that, except from 2 cases, all testing among different extraction protocols, because there are also different extraction protocols designated by colors here. In all cases, the conversion rate for single-stranded library was higher. That means that more molecules ended up being converted into a DNA library. And that's probably because there are certain purification steps during the double-stranded library protocol, and you lose quite a lot of molecules during those purification steps. Well, by having the single-stranded immobilized in the single-stranded library case, immobilized on the beads, you can be sure that you will retain those molecules in the next steps downstream in your method. So another result from the same study. So I said about length distribution before when we were talking about the extraction. So this plot includes also the comparison with library protocols among extraction methods. So what we can see is that within double-stranded, within library methods, the aDNA method for extraction always retrieves shorter fragments. So this is what was expected to do, and that is what was designed for in the first place. So, and we can also see that between library protocols, the combination of single-stranded library and aDNA extraction leads to the retrieval of shorter fragments, which is interesting. So we have a cumulative effect here. Again, this is the last graph of the presentation, and we see the cumulative effect of using combinations of different library and extraction protocols per sample. First is the sample code, the prefix, and then the extraction method, R for Roland, D for aDNA, C for the combination of the two, and then double-stranded or single-stranded library. So the single-stranded libraries are with the blue, and the double-stranded libraries are with the reds. So what we can see here is that there's a great difference in fragment lengths recovered. So when using aDNA with single-stranded library, in most cases we recover more short fragments for a given sample. And we can also see that there's likely more variation between methods rather than samples. So we can see like a fairly consistency among samples for the same methods. So to sum things up, we saw that there are several methods to solve the problems of low endogenous DNA. There's a variety of choices between choosing right samples or performing CT scanning, treating the sample prior to extraction. All these have the potential to lower the final cost of the sequencing, and by basically lowering the contaminant, the amount of the contaminants in your DNA samples. And we also saw that optimized methods such as the aDNA extraction method or the single-strand library have the potential to give better results, meaning retrieving shorter fragments from ancient samples. So this basically opens a new window for studies outside of the permafrost region, and also pushes the time of the time limits further back in time. So we can, by applying these methods, we can actually have the possibility to study species that died far back in time, or they have lived in habitats that were like outside of the permafrost. And this is actually what we do. So we have been using these methods, both the doubling and the single strand library in our lab, and they have performed quite well. We've been applying it to several studies in species that lived mainly in temperate environments. So with the development of methods, we have traveled a long way the last 40 years, I can say. So just to give you an example, to just go back to where we started. So we started with this, which is the quagga, the sequencing of 229 base pairs of mitochondrial genome. This species was extinct fairly recently in 1883, and then within less than 40 years we ended up with this. So this is the latest genome sequencing of a mammoth that has been older. The sample has been older than 1 million years old. So this is quite remarkable what we have achieved with this method development. And some final thoughts about ancient DNA research. I think at the moment we're at a very high level in terms of lab methods. Some suggestions might be maybe study further the fragmentation process. So this is going to give us a very good overview of how far back in time we can go, and how early within the Pleistocene, of course, we can actually study populations. Also, the combined effect of different library extractions and library and extractions would be something interesting to check further. Maybe we will, in the future, we'll come up with certain fixed methods for certain samples deriving from certain environments. So also the exploration of pre-extraction methods would be something to consider, along with post-library capture protocols that I haven't touched upon. This is a very hot topic in also increasing the ancient DNA the endogenous content. And lastly, I think that actually the new computational analysis will bring further development in the field. Maybe meaningful ways of utilizing information from sorted reads that we can retrieve with our library and extraction methods, or more accurate mapping algorithms and such. So that, I hope this presentation was interesting, and I would like to thank you for your attention.